episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show with another fascinating guest involved in creating a better tomorrow for many people. Today on the show, we have the honor of being joined by Dr. Mark Tester, who is Professor of Plant Science and Associate Director of the Center for Desert Agriculture in the Biologic and Environmental Sciences and Engineering Division of King Abdullah University of Science and Technology in Saudi Arabia. Prior to joining King Abdullah University in February of 2013, Dr. Tester was a professor of plant physiology at the University of Adelaide and at the Australian Centre for Plant Functional Genomics from 2009, 2013. He has his PhD from University of Cambridge in plant sciences. And the aim of Professor Tester's research program is to ultimately elucidate the molecular genetic mechanisms that enable certain plants to thrive in extremely suboptimal conditions, including those of high salinity, high temperature, and ultimately deliver outputs in various economically viable systems such as barley, rice, tomatoes, quinoa. A short-term applied application of this program is ultimately to modify crop plants in order to increase their productivity in conditions of challenging stress, with consequent improvements in the yield of these various crops in Saudi Arabia, the region, and globally. A larger aspiration of this is ultimately to unlock seawater as a very important resource by developing these new economically viable agricultural systems where salt-tolerant crops are irrigated and partially desalinized water or brackish groundwater. And there has been a company, Red Sea Farms, that has been established to facilitate and translate the delivery of these products. And Dr. Tester is a co-founder and chief scientific officer of Red Sea Farms. Dr. Mark Tester, thanks so much for taking the time to come on the show today.
Thanks for inviting me. It's an honor to be here.
It's a fascinating topic. Before we go into it, I'd love, as we typically do, to start off by handing you the floor for a few minutes just to further introduce yourself. If we can learn a little bit about you, everything from where you grew up, when you first got interested in plant sciences, plant biotechnology. I think that'll lay the groundwork for a lot of what we're going to be discussing.
Yeah, okay, that's fair enough. I mean, I didn't know I was a plant guy, but I am. And I think quite a bit of this is actually genetic through my parents, and they're, without much education, they would be exposed to plants through gardening and so on. And I think they're very good at that. And then I had the privilege, the opportunity, as society progresses through the generations. So I had a chance to go to university, and that was. I went to university doing science. I knew a sciencey sort of nerdy guy, but I didn't know I was a plant guy until I was just choosing biology as my fourth subject as a minor, really, at in University of Adelaide in Australia, and I loved it. Took to it naturally, and took to biology naturally, and then more particularly took to plant sciences, and it was great, and it was something which I just enjoyed in the raw, and then you get a positive feedback. You enjoy it, so you work hard at it, and then you're successful, so then you enjoy it more. So it's as simple as that, really. And I wanted to do a PhD. Well, actually, I was very interested in ecology and understanding systems and relationships between plants and their environment through time. And it's actually a very complex subject. And there was the ecology professor at Adelaide. He was really good. And he said to me, no, don't do a PhD with me. A, travel, and B, get more training in fundamental plant science and then come back to the system. It was very self-effacing advice, and it was very good advice. And I was lucky to get a scholarship and admission to Cambridge University. I probably went a bit too far in that I ended up doing a PhD. It was in cell biology and doing electrophysiology and studying transport of ions and the electric currents which were generated during the process of the movement of ions across the membrane from the outside to the inside of the cell. It's a long way from an ecosystem, and it's taken me 30 years, but I'm back at the system level now. But it was good. I had good advice, and it was, to not just do 100% what you enjoy, but also get some training and some rigor in your ability to think and analyze, some fundamental knowledge, and then use that, build on that to go to more complex systems. I think it's sort of like people love jazz music, want to play jazz music, but actually classical training is extremely good for becoming a good jazz musician. And I think, yes, training in physiology, at least for me, sure it doesn't work for everybody, but for me, this training in cell biology and physiology has led me to be able to go back towards the system level of work, which we pretty much are now. I mean, my lab now spans quite a breadth of activity, but from the molecular to the system, but that's fairly unusual, I think, in science. But I think we get away with it partly because we've got really good collaborators and colleagues, a very good environment for being able to do quite a few different things, because we're very well resourced. And also we get away with it, I think, because we are focusing on one sort of biological problem of salinity stress and mechanisms of salinity tolerance. It's a little bit of random commentary on my random journey.
No, it's quite fascinating. And when you talk about sort of the systems level thinking, and obviously we'll drill down and we'll get into sort of some of the genetics and the different mechanisms. But the thing I was just wondering from the beginning here, and obviously we live on this planet. I forget, we have a couple 100,000 members of the plant kingdom. We consume a very small amount of those. And I was just off the top was wondering, when it comes to some of these what I'll call sort of extremophilic properties of plants, whether it be salinity or heat or frost resistance or whatever. Are these common in many of the other species that live on this planet, and we just don't think of them, or we've maybe over time bred some of those out to make our corn sweeter and our barley whatever, because you think about it, these plants have been around for a billion years or so now, and they've obviously experienced ice ages and floods and all sorts of other stuff that puts these unique situations, these abiotic stresses, in their way. Are these properties quite common and we just don't see them because we're not around those plants in our daily lives?
Yeah, I mean, I think there are organisms that occupy most niches, most environments on the planet, but often the adaptations to the more extreme of those environments, I mean, it does come with a significant cost, whether it be in terms of the gross morphology or the anatomy or the physiological and cell biological activities and the energies that are required for those. So yeah, there's plants out there for most niches. There are limits, especially around water, and salinity, but there are limits, of course, that are determined by the combination of the physics, chemistry, basic cell biology, basic molecular biology. DNA melts, membranes melt. Water potential is determined by the salt and solutes that are dissolved in that water to make things just too difficult in some environments. But there is a lot of life in a lot of niches, but it comes with a cost. Sure. And for those three plants that provide 40% of at least our energy needs, so rice, wheat, and maize, those three grasses that produce a grain which we can store very conveniently dry. It has a fairly high energy density, which means you don't need huge volumes to store them. They've been developed to be highly productive in fairly reasonable habitats. And I think it's a fantasy to think that we can get those frontline crops and have them grow in very hot or very dry or very salty environments. There are limits and compromises that would have to be taken, which then pulls them out from being a frontline crop that's providing such a massive amount of starch and protein for the human population. So I think when we're talking about some of the processes that we're studying, I mean, I have to admit, for most of my career, I've worked with the paradigm that we've got to produce more food, and there's more food needed on the planet, and we're all going to starve to death if we don't have more starch and protein provision. And I'm not sure that's completely correct. I think plant science, agronomy, the soil scientists, people who are managing the food systems, so food processors and transporters and so on, have built a pretty amazing system. And we now have more people going to bed obese than hungry. We're not going, there are still people with insufficient nutrients, right, but not energy. So we're providing enough carbohydrate for the planet. And in fact, especially if we just reduced by, maybe 50%, if we reduced by half the waste that's also blighting our modern existence, the waste of food that's being produced. Absolutely. If we halve that, we're already producing enough carbohydrate and protein to feed the planet in 2050, not even just today. It's an extraordinary analysis. It's done by the Eat Lancet Commission, published a year and a half ago. And this analysis I found very influential in my thinking. I was also worrying. I mean, I always have been thinking about the degradation of the ecosystem, the environment on the planet. That's always been a concern for me since I was a student at university, and that CO2 levels are rising, water resources are being overexploited, and natural ecosystems are being massively fragmented and destroyed. These are huge issues, but I say that until relatively recently, I have to confess, I was thinking more about, yes, getting plants to grow on salty water and increasing basic food production. And my thinking's changed quite a lot. And I think now much more about the utility of our research on trying to get plants to grow more with low quality water as being a method for reducing the pressures on the high quality water.
Got it.
And I gave you the food supply context, but there's also a very alarming environmental context. So our food system, it occupies half the land that humans occupy, uses half the land that humans occupy. It's the second biggest emitter of greenhouse gases, second or third, depending how you cut the cake and analyze it. But it's a very, very high emission of greenhouse gases from agricultural activity. And of all the fresh water that humans use, and humans don't really use very much salty water at all, but of all the fresh water that humans use, it's about 70%. 2/3 to 3/4 of all the fresh water we use is used for agriculture. Amazing. And half of that water comes from underground. And every aquifer that's being used, every major aquifer that's being used for producing that water, for producing a lot of the world's food, every one is being depleted. So we're actually unsustainably mining water reserves, underground water reserves, to produce a significant fraction of the food that we're eating and that we need to increase in amount. I mean, we still need to increase food supplied, especially for improving human nutrition, iron deficiency, vitamin A deficiency, and so on. Really needs needs to be addressed, massively important issue. But improving the sustainability of our food production systems is something which really has been, really grabbing my attention much more, rather than the absolute need to produce more food. I'm quite happy with that. If we are able to substitute a lot of the fresh water that we use with salty water or other water resources that we don't currently use, if we're able to do that and reduce the pressure on these currently unsustainably exploited groundwater reserves, I think that's quite an important dimension to our food production systems.
Absolutely. So moving into that domain, obviously, your group is focused on salinity tolerance, thinking of these brackish water resources, saltwater resources. You're using a variety of different genetic approaches to understand this. Can you talk a little bit about what is involved in some of these or behind some of these mechanisms of salinity tolerance? Are these mechanisms that allow a plant to get rid of salt, hold on to it without dying, increase stress responses? Like what's I know I drink some salt, some ocean water, and I die pretty fast. What are some of the things that you have to look for into your genetic understanding of what's going on?
Yeah, no, there's that's a very good question. I'm only smiling because I've just finished giving a plant physiology lecture course, a level 200 lecture course on plant physiology, and the last 2 lectures I did was on mechanisms of salinity tolerance. So I can give you about 3 hours now on.
I got a little time.
I mean, it's actually quite. There are many ways that salinity causes problems for the plants, and many ways that the plants try to reduce the toxicity, reduce the inhibition of their growth from being in a saline soil or being irrigated with saline water. I guess one of the ways you can analyze this thinking was mainly developed by Rana Munns, a plant physiologist who worked on salinity tolerance of wheat in Australia for many years. Okay. And she developed some of this thinking and then asked me to collaborate with her, and we wrote a review about 10 years ago, which defines quite a bit of this. She had published some of it before, pretty well actually, but this thinking, I just want to acknowledge her intellectual input. Sure. She helps us think about how there are 2 major components to sodium toxicity. One is due to the ions themselves, and the other is due to other factors which aren't to do with the ions. We're still not completely sure exactly what's going on, but it's what we call the iron independent component of sodium toxicity. In this paper, we called it osmotic toxicity. So we have the ionic toxicity and the osmotic toxicity. Sure. I'm not completely sure that name implies that it's to do with the effect of the salt on the water potential, which it may well be, but I think it's more complicated than that. We're not completely sure. But as a corollary of that, plants can have what we would call osmotic tolerance, tolerance to the non-ionic component of sodium toxicity. And then we've got the ionic tolerance. So we have the osmotic tolerance and the ionic tolerance. And the osmotic tolerance, which is actually more complicated. I should start with the ionic one, but anyway, the osmotic tolerance tends to actually show itself very rapidly. So you can, in an experimental situation, you can have the plants growing, and then you can add even relatively modest concentrations of sodium chloride, of salt, to the solution the plants are growing in, or the soil that the plants are growing in. Mm-hmm. And literally within minutes, you can have inhibition of shoot growth, inhibition of leaf elongation, okay. Leaf expansion. So these effects are very, very rapid, and they're clearly involving rapid root to shoot signaling. And there is genetic variation. Some plants suffer more in that very early response than others, and we need to try to identify the genes responsible for that. And it's still a relatively obscure part of this little field. And the other component is the ionic tolerance, and that can be divided into sort of two different components. One, you've already put your finger on it in your question. One is to try to minimize the amount of sodium accumulating in the tissues of the plant. Mm-hmm. So try to either keep the sodium out in the first place or kick it back out. So you try to keep it out, but some will still get in. So then you want to maximize the pumping back out of the salt, and that's down in the roots. And then you've got the shoots, and there's another mechanism which turns out to be quite an important one genetically, which is to minimize the transfer of the sodium from the roots to the shoots. Okay. And to minimize that, because it's the leaves which are primarily sensitive. They're the most sensitive parts of the plant because they're up here in the air. And the air is already quite a tough environment to be out in. It's quite desiccating. There's all sorts of other stresses. And then now some salt's building up. So this leads to some serious problems for the plant. So if they can keep the salt out from the leaves, then that's really good. And then, of course, some salt will get up to the leaves, and then there will be genetic variation in how plants are able to tolerate the salt that they have failed to keep out. So some salt will get up there, and then some plants go and just die with a bit of salt accumulating up in the shoot, like rice and wheat. Neither of those are that tolerant to the salt accumulating in the leaves. Mm. Whereas other plants are able to tolerate quite high concentrations of salt in the leaves. That's really interesting. What genes are in those plants that are missing from the other plants? And I guess there's a fourth step in this whole process. So plants will try to keep the salt out. They'll kick out what they can't keep out. They will then try to keep it out of the shoot. But then there's differences in the ability to tolerate it up in the shoot. And then there's like a final last resort process that plants can do, which is then secreted out onto the surface of the leaf using salt glands, just as we do. Now, we have salt glands because we're trying to get water onto the surface of our skin for that to evaporate to keep us cool. So we use salt, we pump salt out, and then the water follows. And it's the water that we're interested in. The water evaporates and keeps us cool. That's a mechanism for sweating. Right. So in plants, it's sort of a mirror image mechanism. So what plants are doing, using the bulk flow of water out of the leaf to carry salt out onto the surface of the leaf. That's a mechanism for a gland. And so, glands have evolved many, many times, but they're not that common. So you sort of think that there must be quite a significant cost to having glands. So I think of them as an extreme adaptation to extreme salinity. Like mangroves famously have glands, and there are some grasses which can grow in seawater. They have glands. That's really, really interesting. So lots of mechanisms for lots of different sorts of components of toxicity, and lots of genes that are controlling these processes as well.
Very interesting. Very interesting. And then, now going from, so you study a lot of this genetics, and obviously, though, at some point, then you go to the fields. And it was very interesting when I was looking through, quite a few publications, and one of the interesting ones from a couple of years ago, sort of on this front, was from 2019. It was in Frontiers in Plant Science, titled "Unmanned Aerial Vehicle-Based Phenotyping Using Morphometric Spectral Analysis to Quantify Responses of Wild Tomatoes to Salinity Stress." And here, you talk a lot about, okay, so we're translating, we're going to the field, but obviously, if we have hundreds of different varieties of tomatoes, and some are different than others, we have to. This is difficult for Dr. Mark Tester to get out there, and as everyone talk about sort of the use of some of these new technologies, obviously the drones, but other tools you might have in the drone, artificial intelligence, or other tools that really help you with this phenotyping exercise.
Yes, no, that's an excellent question. So, what we have now, okay. From that previous answer, you can tell that salinity tolerance mechanisms are many and quite complex and quite genetically diverse. Yep. And so if we're to understand, how one plant manages to keep growing in a saline condition, another one is failing, trying to understand the genetic basis of that is going to require not just looking at big plant, little plant, but trying to look at a whole range of different traits of features in those two different plants. And then because it's a set of features, it's a series of features which are making the difference between the salt tolerant and the salt sensitive plant. It's not just going to be one gene. If it was one gene, that'd be easy, and we would have done that 50 years ago. Surely. So we've got to measure a whole bunch of traits. Now, to do that, you can't just do it on 2 plants, all right? You've got to do it on hundreds of plants. And the more able you to do it in what we call the target environment, in the field in which the crops would be grown in the future, is always more powerful, because you're less likely to measure something which is then actually irrelevant. So what we do is try to find what I would call a bunch of proxies, which might be indicative of traits that contribute to salinity tolerance, or might even be fairly directly a trait that contributes to salinity tolerance, but it's got to be stuff that you can measure lots. All right, so you can have a whole bunch of people running around the field with rulers and a camera or something like that. That's fine. That's one thing you can do, but we really do have amazing technologies now. So we're really in the middle of a revolution, and it's a technologically driven revolution with genomics, enabling us to sequence hundreds and hundreds of genomes, much more easily than was possible even 3 years ago. And then we also have these what we call phenotyping technologies. So technologies to measure the size, shape, behavior of the plant. And this is using digital cameras. And you can use digital cameras on the back of a tractor, the front of a tractor. You can use digital cameras on a drone, in an air balloon. It doesn't really matter. Drones, I think, are quite good, because I think they sort of hit the middle ground. I mean, you can use satellites as well, of course, I should say. The satellite technology is amazingly powerful now. But let's stick to drones for the moment, and you can put different cameras on the drones, and then you can take them to different fields, and you can put different cameras on the drones, or even fly more than one drone at the same time. And this leads to huge amounts of information coming in using digital imaging technologies. So we've got powerful computers now that can store lots of stuff and analyze lots of stuff. You've got digital imaging technologies that can capture lots of information. And then you mentioned the third component that you need. You need to be able to understand what all that data means. And I actually quite like what's happening now, because I used to think, I reckon I know what's going on. Let's measure this or let's measure that. And now what we can do is use machine learning and and let the computers tell us. So you can give them you can give them a set of plants to learn from, a set of data to learn from, where you're looking and manually measuring, ground truthing, doing all sorts of things like this too, and calibrating, if you like, the equipment and comparing it to yield, for example, or the maintenance of yield in saline conditions relative to non-saline conditions. You can calibrate one set of data, and then the computer's learned, and then you can give them the rest of the field that is too big for you to run around with your ruler and scales, and start to let the computer tell you or predict what's going on, and then tell you what components that they're measuring were important. And then if you can start doing this in hundreds or even thousands of plants, if you've also got the genome sequence of those plants, then you can start doing a massive brute force statistical correlation. I make it sound relatively simple, but it's much more nuanced than that. And some of the smartest people in plant science do this work. And I'm not counting myself in this, but there's people who are what are called statistical geneticists who do this massive brute force correlation to identify the probability that variation in a particular trait that you're measuring is due to variation in a particular part of the genome. And this is getting so powerful now that you can sometimes, if you're lucky, go down to the gene straight away using this brute force statistical approach. But for example, we've got one trait that was measured by a PhD student. She did her work, and now postdoc, and then she finished, and now postdoc is following up, looking at the genes which are in the interval. This is to help look at this is to identify genetic control of the ability of plants to maintain what's called transpiration use efficiency in saline conditions relative to non-saline conditions. And there's 25 genes in that interval.
Wow.
And so this postdoc is using more traditional, what I would call traditional, there's still modern methods of molecular biology and brute force genetics to try to identify, brute force molecular genetics to identify which of the 25 candidate genes is the one that is responsible for, or which has variation that is responsible for variation in maintenance of transpiration use efficiency. That's one real life example that we're in the middle of in the lab at the moment. And maintenance of transpiration use efficiency is a significant contributor to salinity tolerance.
Very interesting.
So if we identify the genes and the versions of the genes, they're called alleles of the genes,
Sure.
Then those can be introgressed into varieties which have got other positive traits, quality traits, or other attributes, heat tolerance, whatever, that is also required. So you can bring in the version of the gene to your primary frontline commercial varieties.
Very interesting. Mark, talk a little bit about Red Sea Farms, if you would, because you're actively teaching. You're a professor at the university. This is a spin out that the King Abdullah organization is also, I think, an investor in. So there's a nice sort of tech transfer ecosystem there. Talk a little bit about what you're doing at the company and some of the goals. I also noticed something on the website about seawater cooling, which is another research aspect. Talk a little bit about that, if you would.
Engineering. So, look, KAUST is established as a university to, be a top research university, reignite scientific excellence in this region, and make significant tangible contributions to the future post-oil economy that is looming ahead of the region and the world. And except it's particularly critical in Saudi Arabia, where so much of the economy has been based on oil. So they really, in Saudi Arabia, really do need to develop a post-oil industry and economy quite rapidly. And so the universities, this is an imperative of the university. And of course, you can contribute in many, many ways, through education of young people, which is possibly actually the most important of all of them. The influence of policy through government and through the big companies. That's another very important way you can contribute to how a university and academic can contribute. And then, of course, there's a lot of outreach and educating people in wider society, and seeing if you can encourage people to, or give people opportunities to change behavior and make decisions which are beneficial to the economy and the environment. And then, of course, university can also license technology to, or do even almost sort of for service research, collaborative research with big companies. That's also a very important thing to do. But there's another way as well, which is to foster entrepreneurial activity and see if research developments, research breakthroughs can be delivered through small companies that can grow to be big companies. And it's a very good mechanism because it's dynamic, and as long as you're willing to take on the chin, quite a high failure rate, then you get, 90% of companies or something like that are not going to make it through, but some do. And then when they do, you've got yourself you can often success breed success as well. So you can have things feeding off. You get a good positive feedback churn to deliver research outputs. And KAUST is, I think it's been really good with this. They've got the means to support, admittedly, so they've got good funds to invest, but they've made those decisions to invest. So there's an environment where I think there's more opportunities for research developments to get through that valley of death and out the other side into a developing company after being a startup for 1, 2, 3 years. So I like it. And I'm doing my stuff with my plant science and understanding salinity tolerance. That's all fine. It's good work, and I like my science. But I'm very lucky to have partnered with an engineer, an agricultural engineer who specializes in water. In fact, I was on his PhD. I mean, we knew each other from the beginning. In fact, we started at KAUST coincidentally, just a couple of weeks apart from each other. Ryan was just, I don't know, he started 3 weeks before me. And we were acquainted with each other. And then he asked me to be on his PhD committee, which was really good. So I got more deeply understanding of his work. And he was working on the use of brackish water for cooling in this environment. Now, many people listening won't perhaps realize, but the environment on the coast here in Saudi Arabia is not only very hot and dry, but it's also really humid. And so this is not a good environment for doing a lot of things like evaporative cooling. And so Ryan was also working with liquid desiccants to try to create an environment where the cooling would be possible. And what you find is that, in fact, during the day, you can get some level of cooling with evaporative cooling, but at night time, you really don't. It can be, even though the heat load is much lower because the sun isn't shining in on your glass box, the greenhouse, but you have 100% humidity. It goes right up to saturation for at night for a significant fraction of the year. And so it's impossible to evaporatively cool. And so Ryan's developed a system using liquid desiccants which you regenerate during the day using solar power. So you regenerate and cool during the day, and then at night you can use to dehumidify the air. prior to cooling it. So it's really, really nice system, and you should get Ryan to explain it better than me. But the principles for Red Sea Farms is that we've got an engineer who's doing smart stuff to enable us to do cooling in difficult environments using seawater. And then I'm the gardener. So I'm the plant guy who is both able to help with growing the plants in the first place, and also, of course, growing them with some reduction in freshwater consumption. And yeah, like a lot of successful companies, you need two complementary skill sets. It's often technical and financial. We happen to have two very complementary technical people. And then fortunately, we've been joined by a financial guy as well, Simon Bryant, and he's amazing in terms of the business development. And Ryan and I focus. I mean, Ryan does a lot. He does a lot of the business development as well. But we've got this nice complementarity with the technical skills. So overall, we can grow salt-tolerant plants in a greenhouse on diluted seawater, not straight seawater by a long way, but it's on diluted seawater in a greenhouse which is cooled using 100% seawater, and where the roof is cleaned using no water, because we're in a dusty environment. If you don't clean that roof a couple of times a week, you get a measurable reduction in yields. And so we end up with a system overall. Here we are, back to systems. We end up with a system overall where, depending on the temperature, humidity, time of year, crop, from 80 to 95% of the fresh water being used is substituted with seawater. I reckon that's pretty good. So if you've got a bag of tomatoes, you go to the supermarket, buy a bag of tomatoes in this type of environment. Usually for 1 kilogram, that's about 4 big tomatoes, which isn't many tomatoes. That requires about 350 litres of water.
Yeah.
And that's whether they're grown in an open field or in a normal evaporatively cooled greenhouse in this region. And so with our technology, you end up with that bag of tomatoes using about maybe 20, 30 litres of water instead of 350. So it's not zero water. And this is where I think we're a little bit different. We're not going around saying we can use 100% seawater.
Right.
We could do that technically, but it wouldn't make any economic sense. And so what we're trying to do is find a sweet spot where the contributions to reducing the environmental footprint of our food are significant, but not so the business doesn't make financial sense. So it's got to be economically viable as well as environmentally viable. That's the only true sustainable. You can do things and make nice movies on the internet and make all sorts of claims, right. But if they don't make economic sense as well as environmental sense, then they're not truly sustainable.
Sure.
And so that's what we've tried to put together as a company, which is, very much driven by the environmental sustainability imperative, but still very strictly ensuring it's also economically viable.
Sure. I was in Jeddah a few years ago, and I can vouch for that that humidity at night, because I was there after during the Eid, and I was hanging out at night. And wow, this is it's a steam room everywhere I went. But
that's right. Why am I sweating so much? It's midnight.
But anyway, side story. But speaking about sort of things making sense, I was looking at a map before of sort of where you're located on the Red Sea, and I just obviously across the way is Eritrea, and I remember a documentary. This had to be about 10 years ago now, about a group that was actually watering the desert. With the hopes of planting species like Salicornia and other sort of already salt tolerant. I don't know whatever transpired with that stuff, but could you give us a few minutes just on your top line thoughts of the greening of the deserts per se versus, I mean, not as an alternative to what you're doing, but is there any complementarity in that type of stuff, and is it useful at all?
Yes, no, sure. No, that sort of work is fine. And you must manage the drainage, of course, so the salts don't build up in the soil. So you have to have the system to be sustainable itself, where you're loading a bunch of salt water into the system every day. So it does have to be managed, through flushing and drainage. But that's, it has to be, again, that's technically possible. It has to be financially viable as well. So no, I'm a strong supporter of developing systems where we can grow plants in seawater. In many ways, that's the ultimate. I think they're going to be moderately limited in scope because of the areas of land which are going to be suitable for growing and managing, growing with very salty water and draining. Having said that, full seawater is a big push, but there's a lot of brackish water in the world. I mean, for every drop of fresh water underground, there's a drop of brackish water as well. So brackish means above what we can use, but below seawater. So in that range, there's a lot of water in that range, and we can, I'm sure, develop systems that could be quite widespread with that type of arrangement. Again, as long as we can manage the drainage water and not mess up the environment even more than we already are, in the quest of trying to do something good. And those Salicornia systems, I think there's a couple of challenges that need to be addressed, but are eminently addressable. And one is ensuring you've got a market for what you're growing. So no market, no business. Right. So they have to be able to sell what they're growing. And that, I think, is quite important. Salicornia is a good plant, though, because it produces an oil-rich seed, which you can squeeze and produces a high-quality oil. It's sort of similar to sunflower oil in its properties. So you can use it as a cooking oil. You can use it as a lubricant oil, apparently. So my engineering colleagues tell me. And I mean, people tout it for biofuels. I mean, I wouldn't really go there because you'd have to grow so much to make any really significant contribution. And fuel is, but airplane fuel is definitely a long-term challenge for the planet. There's no doubt. And I guess this could make a contribution towards that. Quantitatively, I'm not so sure it's going to be that significant a contribution, but it's all fine. It's better than nothing. I think the other problem with the Salicornia, just growing Salicornia, is it's just a wild plant. It's an unimproved or barely improved wild plant. It's sort of like going down to the edge of the highway at this time of year and collecting a whole bunch of grass seeds from the edge of the highway and making a loaf of bread from it and then expecting to make a living from that.
Got it.
All right. It's just, you're growing a wild plant. It's like going out and harvesting a wild plant that's of moderate to low value. And that's not a good business model. And so, in fact, in the university, look back to KAUST, we've got I'm the associate director. The director is Rod Wing, a very good plant genomics guy. We lead what's called a Centre for Desert Agriculture. And that Centre for Desert Agriculture has got two primary research foci. One is dates and date palms, which is an iconic and very culturally important plant for this region. The other research focus is domestication, neo-domestication. And one of the main things we're doing is getting plants which can grow in very saline conditions, like seawater even, and domesticating them, getting them to become a commercially viable crop, changing their properties. So we will be able to get that field in Eritrea and grow a field of Salicornia and be able to make it an economically viable business, like really deeply economically viable. So you can get bigger grain, higher yield, better quality grain and so on. you have to have genetic improvement,
Mm-hmm.
hand in glove with the development of the agronomic system. You need both of these things. And the guys who were doing that field in Eritrea, similar, they did fields in Arizona and in Mexico as well, in the Sonora or Baja, and they in northwestern Mexico. Pretty much just in inverted commas using the wild plants. That's a tough call. I think you have to genetically improve it as well. But I think it's an excellent aspiration, and I think we should be domesticating plants that can grow in seawater, and it will be a contribution. I think it could be even a significant contribution to global agriculture. Yeah.
Excellent. Mark, final question. You mentioned your business partners in Red Sea Farms. You mentioned your family early on. Take a couple of minutes just to mention, shout out to anybody else. Obviously, people at KAUST, people from Australia from the past, anyone else that has been really instrumental and important for you as you've been progressing and developing these issues over the years.
Yeah, I mean, we all need mentors, guidance, advice, teaching. Our job, as an individual is to learn, work hard, and try to have that positive feedback loops of excelling, enjoying, and achieving something. And sure, along the way, you need it's countless people. At the very start, I was a very raw boy back in Australia as an undergraduate, and there was a husband and wife, Andrew and Sally Smith. They were fantastic and pointed me on the way, taught me how to write, and really did teach me a lot just way back in my undergraduate days. Then I went to Cambridge to do a PhD with an amazing woman, Enid McRobbie, and she really taught me to think analytically and quantitatively. So she had a degree in physics originally, and was then applying that to plant systems, and that's great. So if you're taught to think tightly, rigorously, quantitatively, that really can be very, very helpful, and that's been very helpful through my career. And I've got, it's just been a wonderful journey, really, in many ways. So I think the other sort of more broadly, I think the students are the best thing. You have good colleagues and all of that, but students are learning, and then they ask good questions, and having bright students who really outsmart you, I love that. And so I teach, but I learn while I'm teaching, and that's some of the most rewarding parts of life, actually. So I've had some outstandingly good students who ask good questions, and I can probably thank them more than anybody, actually, for they keep you young, keep you questioning yourself, keep you questioning your science, and the act of preparing to teach really helps you ask questions as well. So I'm not a guy who, although I've had a lot of time where I have not done huge amounts of teaching, a lot of, I think, professors they get burdened by teaching because it's just too much teaching. I think I've been really lucky to have a good balance in teaching, and between teaching and research. So I've been able to be lucky to learn from teaching and then have enough juice in the battery time to be able to do some research from that teaching. But I do like that balance between teaching and research. I don't think they should be separated.
Sure. Well, Mark, it's a fascinating journey. I'm going to look forward to you continuing on this path. There's just so much potential to it all, and really rooting you on from here. Hopefully I'll be able to meet you face to face one day next time I get over there. But the really outstanding work for everybody that's going to be listening to this episode on the podcast or watching on the YouTube channel. You've been spending time with Dr. Mark Tester, Professor of Plant Science, Associate Director at the Center for Desert Agriculture, King Abdullah University of Science and Technology in Saudi Arabia, also co-founder and Chief Scientific Officer of Red Sea Farms. Mark, it was a great time talking to you. I want to thank you for taking the time out of your schedule to do this. I want to thank you for everything you're doing. And as we say on this show, thank you for helping to create a better tomorrow through what you're doing. Really inspiring stuff.
Thank you very much, Ira. Thank you for the invitation, the opportunity to talk about this. It's been really great.